All right, so this is the Neuro 481 lab course, coronavirus edition. So due to this SARS-CoV-2 virus, we've had to move everything online. So maybe introverts and night owls and germaphobes are celebrating this move, but this whole COVID-19 pandemic really is a serious risk for even healthy people, and that's why things have gone in this sort of unprecedented chain of events. Since this is meant to be a lab course, I hope this doesn't compromise the applications and more interactive components of the material that we're learning. But I think we're able to do this, and I think everything will go well. So the classic joke is that every disaster movie starts with the government ignoring scientists, and then it's too late. And sure enough, this disease right now has a massively devastating potential, which we've already started to see in other countries where hospital systems and resources have been completely overwhelmed with patients dying in the hallways and things like that. So we're obviously trying to avoid that at all costs. Only a few times in my life have I ever had to bronch patients in the ICU, and only once was it a viral pneumonia patient with a secondary bacterial infection. But just watching that one person die, despite every conceivable effort at ventilatory mechanics, was awful. And I can't imagine hundreds of these patients filling the hospital. This whole quarantine makes sort of a giant, unprecedented research experiment in epidemiologic transmission. And so I hope you'll think back to the stats lab that we did, and about relative risk factors and bell curves and comparisons of significance based on different treatments or based on different preventative measures and all that sort of stuff, and kind of relate that to these current events. Along with that, I was just looking at this the other day, there's actually an interesting model that you can get on GitHub from Johns Hopkins, from the Center of System Science and Engineering. And they actually reported on it in the Washington Post as well. It's a nice model showing how social distancing is really effective and dramatically limits the spread of disease, whereas other things like localized forced quarantines are not particularly effective for various reasons, but that's another topic. So anyway, if we can't cover everything we normally would in lab, we can at least try to cover the important information and application of the more interesting topics. So as usual, I like to start with some clinical applications of questions that relate the material to actual use in the real world. And then we'll kind of go in deeper theory and come back to the real world again, hopefully. So these questions are just from my neurosurgery board review book, and they show that even in that, you need to understand some basics of electroencephalography. So the first question here shows this sort of global 2 to 3 hertz frequency wave in a 10-year-old girl. This is the classic absence seizure, which you probably learned on your other courses, and this would include the classic staring spells as the primary symptom. And these are types of seizures that don't really have a post-ictal phase. So on the next one, it asks how to normalize this rhythm. And you can see here that in a couple of the leads, there are these really noisy high-frequency waves. And this is actually kind of a tricky problem. But what's going on here is that this is actually some sort of intracranial injury where the bone flap has been removed. So the EEG that you see is actually a fairly normal EEG. I don't think there's any seizure going on here. It's just more that the bone itself acts as a low-pass filter, which means that high frequencies, which would normally get filtered out, are not being filtered out. So any lead that is over where the bone flap is missing is going to have these high-frequency waves, which is essentially the unfiltered neural activity closer to the surface of the cortex. So the answer for this is just to normalize the EEG. We would do a surgical procedure to put the bone flap back on. The next EEG asks what abnormalities are most consistent with this EEG. Now this one, when I first looked at it, I thought it was the sort of classic sawtooth waves of Creutzfeldt-Jakob disease, but it's actually triphasic waves here, which you can see goes up, down, up, down, up. And that's more characteristic of a global metabolic abnormality, especially hepatic encephalopathy, where you can get an ammonia buildup and pathological neural activity. Now going to the next slide, you can see the standardized positions of the electrode leads for an EEG. you can see that over the FP2 to F8, that's sort of this right temporal lobe where the arrow is pointing. That's where a seizure appears to begin, and then it spreads throughout the rest of the brain. So this is most likely a temporal lobe epilepsy, and the most common reason for this is a mesial temporal sclerosis or hippocampal sclerosis. And so this is on the right side of the patient, unlike most images in clinical medicine, which have the patient's left on your right. So when you look at this image, it's actually looking down onto the top of the head. So the right side of the image is actually your right, opposite of most clinical images, just FYI. Okay, on the next set of problems in the middle, we can see a similar thing going on. It asks what is most consistent with what diagnosis. And again, this is showing a seizure beginning in the F4 lead. So this suggests some sort of right frontal tumor as a likely diagnosis. Now, on the left, we have an EEG, and it says, this is most consistent with what? You can see that we don't really see any EEG activity at all, but we are picking up something else. We're picking up some biosignal. It's the electrocardiogram with a frequency of about 60 beats per minute. So this seems consistent with a lethal closed head injury where there's no EEG activity, but the heart is still beating. And this, of course, brings up the topic of brain death. I actually don't call it that. Instead, I'd call it death by neurological criteria. And we could talk a long time about this, but first of all, you don't classify someone as brain dead just based on the absence of an EEG signal. That's sort of just an extra measure that helps you qualify your diagnosis. Death by neurological criteria is basically saying you're dead by the criteria of the fact that your nervous system no longer works. So you lose all brainstem reflexes, the ability to ventilate, and so on. But brain death frequently gets misconstrued in the media, but that's a basic overview. Okay. Now, the last one is this EEG showing labels here in the middle of the time sequence of eyes closed. And you can see this rapid sort of 8 to 10 hertz frequency. So this would be the alpha rhythm, which is what happens in the vast majority of people when they close their eyes. So altogether, these are just some clinical images or examples of EEG. And there are a lot of other applications as well, which we'll see briefly. Okay, first, let's go back to the basics of electricity. So for those of you who might not have taken physics in a long time, that's okay. This is just kind of a nice opportunity to review some basics. So I like to explain electricity with a simple analogy of a waterfall. With a waterfall, there's a certain height that the water falls over, and there is a certain amount of water that flows over the waterfall. The height of the waterfall is essentially like voltage, and the amount of water flowing over the waterfall is essentially like current. In fact, we use the same word, current, for the water and for electricity as the number of electrons that move through an area per second. So, as you can imagine, there can be really tall waterfalls with only a tiny little trickle over them, which would represent a high voltage, low current waterfall. Or you can get really short waterfalls that have a ton of water flowing over them. For example, a Mississippi levee, and when it breaks, you have millions of gallons a minute flowing over the levee, but it's only falling a short distance. So the values of voltage and current, they can be related by the classic formula of voltage equals current times resistance, but there's a lot of factors that go into how the real-life electrical systems function. So in terms of neurons, a neuron is a really interesting, fascinating structure. It's this tiny little sphere of life, and yet somehow it can generate up to 100 millivolts. So this tiny living cell somehow generates this huge potential relative to its size, and it's like a really tall waterfall. But the amount of current that actually flows across the membrane during something like an action potential is actually extraordinarily tiny, something like a few hundred ions moving back and forth across the membrane. So you can mathematically model that and even measure it, which is cool, but in terms of a descriptive model, this is like a really tall waterfall with just a tiny trickle over it. And the information being conveyed among our neurons is through this sort of field potentials, these potentials. It's fascinating if you think about it. It's also really good that the system was set up this way, because if we were doing it with lots of currents flowing through something like a microprocessor where you had currents that were like transistors indicating ones and zeros. Now there is some aspect of neural networks that does function like microchips, but there are a lot of differences too. So if we think of this, if information was being sent as these currents through these impulses, the brain would overheat immediately. The amount of information flowing through currents would melt membranes and make this giant slop of oatmeal in our heads really quickly. So the fact that you can convey information as these stored voltage potentials is actually an incredibly efficient way of doing things. It's actually fascinating to think our brain in terms of sizing is really simultaneously as small as it can be to be as intelligent as we are, yet also as large as it can possibly be without being too massive and generating too much heat that it would melt down the system. So anyway, there are a lot of other things too that can carry information and field potentials, and who knows, maybe even the field potentials of gravitational bodies and their oscillations and the rotations of planets around suns and stars around the galactic centers and galaxies around these sort of tendrils of superclusters and things. So who knows? Maybe the galaxy itself is in some way intelligent. Obviously some kind of sci-fi thought there, but it's just interesting the ways that different information can be stored and carried and transmitted. So anyway, moving on. A couple important points to cover here. So it's okay if you don't remember all this. We'll try to go through it, and fortunately on a video at least you can rewind if you didn't understand something instead of frantically taking notes. So hopefully this video actually helps you guys. So let's just remember that when you have two resistors in series through R1 and R2, let's call them, the total resistance through that wire, through that circuit, is going to just be the sum of R1 plus R2. Now, if you have two resistors in parallel, it's basically going to be the inverse. 1 over R1 plus 1 over R2 will equal 1 over the total resistance. And if you do that algebra, you can actually rearrange this and solve for total resistance, and it'll be this other formula, R1 times R2, over the sum of R1 plus R2. Either formula is fine. They're the same formula. It's basically just like the Worker theorem from your college admission tests, where they ask you if Bob can paint a fence in 10 minutes and Joe can paint the fence in 20 minutes, then how fast do they paint the total fence if they paint it together? So you just use the same formula, 1 over Bob's time plus 1 over Joe's time equals 1 over the total time if they work together. And if you don't remember that, no worries, you're here now. So anyway, when we record from a neuron, we want the signal to faithfully represent the actual thing going on in the neuron. And if we measure voltage potential across a neuron membrane, we're going to call that voltage V in. And that is the voltage that is actually present in the neuron. But when we record through our system, it's going to introduce imperfections, and we have to understand the mechanics of how our system is altering the perceived signal. So no matter what you record through, it's going to have some level of resistance, whether that electrode is a glass pipette or a metal electrode. It introduces some amount of resistance into the recording. So I'm just going to call that resistance R2. You'll see why in a minute. And then we're going to get some signal out of our device. We're going to get a V out signal. V out is the actual signal that we're recording. We have to measure this with reference to some ground, some relative zero value. So it doesn't have to be a literal ground. It can be a digital ground, but that's what a voltage is. It's just like the waterfall analogy. It's the height difference between where the waterfall falls from and where it goes to the ground state, right? So that resistance to ground is going to be R1. So now we have two different resistances, R1 and R2. And it turns out that just in general, the V out is equal to the V in times R1 divided by the sum of R1 plus R2. This is called the voltage divider equation, and this is something you should know. So our goal here is to make the voltage out, the voltage that we're measuring, match the voltage that's coming into our system, the actual voltage of the neuron. So you can see in this formula, the way that we can equalize V out with V in is simply to make R1 much, much greater than R2. So when we do that, that R1 trends towards infinity as a limit. And then you can see in the equation that the R1s will effectively cancel out in their limit, And since R1 is so big, it really becomes irrelevant. The limit of R1 approaching infinity makes that value of R2 essentially irrelevant to the equation, and this is called a high impedance system, meaning the R1 has a high impedance. Impedance is just like resistance, but impedance implies an oscillating signal, whereas resistance is typically used with a DC voltage type signal. So our neuron is oscillating, so we're just going to use the word impedance. And these are the types of systems that we use in electrophysiology when we record from neurons. Now, you can measure neuron potentials directly, either with a glass electrode or metal electrode, and each one introduces unique characteristics into your system. So let's talk quickly about this. You can make a little glass electrode by melting a pipette over a flame and pull it apart to make this tiny little string of glass that has a tiny little opening, and you can insert that little opening under a microscope into a neuron. And just like with axons, the diameter of the axon relates to its resistance, and same thing here. The wider diameter axons have a lower resistance, and a wider diameter pipette will have a lower resistance. If it's a smaller diameter pipette, that'll have a greater resistance, and the greatest resistance is where it's smallest, at the tip of the electrode. So that's something that we're going to call tip resistance, and we're going to label it R2. Now, in the pipette, we're going to have some electrolyte solution that matches the intracellular features of the neuron. And most of the resistance coming into this electrode is going to be at the tip, like I said. And we want this resistance to be something like an order of magnitude higher than the resistance through the neuron itself. So in real-life scenarios, this R2 resistance at the tip tends to be around 5 to 10 megaohms, whereas the resistance in the neuron tends to be about 1 megaohm. Now, there's a capacitance across the glass wall of the electrode as well, just like you've talked about capacitance across neuron membranes. The glass material is a dielectric, meaning it's resistive to electric flow. It's an insulating material. And if you remember, a capacitive flow of electricity occurs when there's a change in voltage, not just an applied voltage, but a change in voltage. So the equation for that is just current equals capacitance times the change in voltage over the change in time. Now, this means that whenever the voltage changes, current flows. And if there's no change in voltage, or if the neuron is just sitting at its resting membrane potential, then there's no capacitive current flow at all. So the insulative glass material is going to have this capacitive current flow that we call C2. And that sort of inline capacitance is going to slow down the frequency response, and we'll see why here in a second or so. In our model, the voltage is coming in from the neuron, goes through R2, and then it can either go to the voltage out that we measure, or it can cross C2 and go to ground, ground being the ground state of the electrolyte solution surrounding the neuron or the extracellular fluid. So just remember that if this mathematics doesn't make sense to you, it's okay. You don't have to remember calculus and things like that. The concepts here are relatively simple and straightforward. Hopefully they make sense to you, but feel free to ask questions if they don't. So a capacitor can also be thought of essentially like a frequency-dependent resistor. You can just talk through these circuits logically without any mathematics, like high frequencies will tend to cross capacitors more easily, and low frequencies will tend to cross resistors more easily, and vice versa. So if we do what's called a spectral analysis, where we show the power of the measured signal at different frequencies, the signal will stay at its original power until it starts to reach this cutoff frequency that's introduced by the capacitance. And the cutoff frequency is shown when signal power is reduced to 50% of its original, let's say. So when this occurs when the frequency is equal to 1 over R2 times C2 times 2 pi, which comes from these cool electrical engineering equations. There's a lot of mathematics behind it that you don't have to understand. But if we draw these results on a logarithmic scale, it will look like what you see here, where the signal drops off slowly as we reach higher frequencies. And what this means is that eventually we're going to reach certain frequencies where we can where we get no signal out of a recording, even though the high frequencies could be occurring. So if we haven't built the system properly, we may think that the neuron is not firing when it's really firing quickly, and we're just not recording it through our system. And we get another interesting effect with metal electrodes. So you should compare metal electrodes to glass electrodes. With the metal, we have lower tip resistance than glass, but there's still a resistance at the tip. And a metal electrode is also coated along the majority of its body with some sort of insulative material like parylene or other polymers. And the capacitive effect will also occur on metal electrodes just like it did with the glass across that polymer coating. In addition, it turns out that just the way that metal is, by its lattice structure or whatever, it introduces a little bit of capacitance at the tip. So when we draw a model, this voltage in will have to go both through a capacitor, which we'll call C1, and also through a resistor, which we're still going to call R2, the tip resistance. Now, we also might lose some signal across the capacitive effects of the insulative coating, which we're all going to ground, so we'll call that C2, and this creates an interesting effect where not only are you cutting off frequencies at the high end, which is still the cutoff frequency of 1 over 2 pi times R2 times C2, but we're also going to lose signal. If the signal is too low a frequency and it's not going to cross the first capacitor C1 at the tip, that means we're going to lose some signal at low frequencies as well. So this creates this sort of interesting band pass filter where we only get frequencies passing through at a certain band of frequencies. So the place where frequencies will start to reach 50% of max signal is 1 over R1 times C1 times 2 pi. So this is all a phenomenon of RC circuits. You've probably heard of RC circuits. These are used in all sorts of electronics. This is what helps us tune in to a certain station on our radio. You create a band pass that allows a specific frequency to pass through your radio. But all you really need to know out of all this information is how to model the two different types of electrodes. So just understanding that and the ultimate differences that arise is what's important. And so just as a summary, with metal electrodes, you'll get a bandpass filter, and with glass electrodes, you'll get a low-pass filter where you cut off the high frequencies. Now, the EEG can record electrical activity from the brain, and if we draw the brain, here we have the cortex of the brain, which will have white matter below that, and above cortex, cerebral spinal fluid, meninges, including the dura, and then skull and skin. And we have all these things that are attenuating our signal. So even though a neuron can generate 1 to 100 millivolts, at the skin where we measure it, it's attenuated down to about 1 to 100 microvolts. And so because of that, we're going to want to amplify the signal that we record. So usually we'll do about 10,000 times amplification, which puts our signal back up to about 10 millivolts to a volt. And the EEG is recording synaptic activity of neurons. I wouldn't say it's recording neuronal activity, except maybe in pathological events like seizures or sleep spikes or synchronous activity where you purposefully trigger with a stimulus like a visual evoked potential or an auditory evoked potential. But it's not individual cellular activity we're recording. It's the sum of synaptic inputs. So if we draw this neuron here, like a pyramidal neuron in the cortex, for example, it's going to have multiple inputs, like inhibitory inputs, GABA neurotransmitter from basal ganglia, for example, or it's going to have stimulatory inputs like acetylcholine or glutamate from the thalamus, for example. And all of these are going to sum up on the dendrites as either excitatory postsynaptic potentials or inhibitory postsynaptic potentials, EPSPs and IPSPs. And once they reach threshold, a certain threshold depending on the neuron, then voltage-activated channels can open and trigger action potentials. But we're just talking about the sum of synaptic activity here. And so I should mention a little history here. I always like to introduce a little history of how we know what we know and everything. So the history of EEG is actually pretty fascinating. A quick excursion of history here. So there was this guy named Alfred Loomis. He actually is credited with discovering EEG, or at least some components of EEG. He was the son of a physician and studied science at Yale, and kind of disappointed his family in a way by not pursuing the physician pathway. And then when World War I came, he joined the military, and while in the military, he started research on radar and ultrasound and things like that, which were just starting to be developed. And after World War I, he got out of that, ended up studying law at Harvard, and randomly enough started a hedge fund on Wall Street. And even though he used some methods today that we might consider insider trading, back then they weren't illegal, and he actually was extremely successful and wealthy on Wall Street. And he actually sold it all off and converted it to gold and cash right before the crash of the stock market in 1929. And then when everything crashed, he bought back up all these stocks and things that were well below their intrinsic value. And he became one of the richest men in the world. And he could have just retired and had a nice life, I guess. But he was fascinated still with science. And about this time, a lot of other stuff was going on that would eventually lead to World War II. And Alfred Loomis, he decided to set up a research lab in his own mansion in New York, and he invited scientists from all over the world, people that we know well today, including Albert Einstein, Werner Heisenberg, Niels Bohr, Enrico Fermi, and all these other great minds, and he invited them there to share ideas and collaborate. And about this time, scientists had just figured out through this whole idea of protons and electrons and neutrons. And Fermi, especially, had hypothesized that neutrons could be used to split protons apart, because protons, they're all positively charged, they repel each other, and so that didn't work too well. But he suspected that it could be done with neutron beams, and the Germans were already working on this, and I think the neutron beam had already been demonstrated, in fact, by some German scientists under Hitler. So Fermi is actually one of the ones who first warned the U.S. about this concept of the atomic bomb, which led to this whole race for building the atomic bomb and the Manhattan Project. And a lot of this went on in Alfred Loomis's mansion in New York. So anyway, Loomis was recording electrical signals as part of his experimentation. He recorded them on his own children, and that's where he discovered the K complex that you guys have learned about in your other classes, this sort of phenomenon when you're falling asleep that triggers the K complex. This whole story is actually interesting because he kept doing research on radar too, making a rad lab that became part of MIT, and that led to the use of microwaves, which basically was a British technology at the time, and that's essentially what won World War II, this British technology to detect incoming German aircraft. So they say classically what won World War II was British technology, American steel, and Russian blood, as they say. So anyway, just sort of a fascinating backstory on these world wars and everything. But EEG, we're going to use these recording electrodes on the scalp. They'll have a resistance, I think, somewhere around 100 kiloohms, and these will go from the skin to a differential amplifier, which we've already talked about. Now, this is important because any recording you do is going to have electrical interference just from mainline currents in the wall or lights above us or things that electrical magnetic interference. So a differential amplifier, as we talked about with our polygraph recordings, it rejects common signals. It rejects the common mode, but allows signals that are different to pass through. And then when we do our operational amplifier, which has the 10,000 times gain, amplifies our signal. And what we want to do here is isolate the signals that we're interested in. So we need some sort of filters that takes things out below the heartbeat range. So anything less than 1 hertz, which is 1 oscillation per second. We can eliminate that because it's not really relevant to EEG as far as we know, at least. And we also don't think there's much significance in electrical signals that are over 100 hertz, and so we can eliminate those. So we simply make a bandpass filter where everything below 100 hertz, like eye movements, muscle movements, they get filtered out of our recording, as well as anything above 100 hertz. So we make a nice little bandpass filter, and we have to remember as well the Nyquist theorem, where we have to sample at least 2 times of our maximum frequency. So we should be sampling at least 200 times a second in order to capture a high-fidelity 100 hertz signal. So in our analog to digital converter, the ADC that you see, we should do at least 200 hertz. And that signal is going to go to the computer. Nowadays, we have software that can do these filters. You don't have to actually program them on big machines anymore. But this signal is going to come out in what we can call a time domain. It comes out in real time. But it's also kind of nice to analyze the different frequencies that compose the signal. So this involves converting a time signal to a frequency domain where we can see how big the different waves are at different frequencies. And by doing this, we can analyze whether there's any clinical association with these different frequencies to pathologies or other research applications. Now, normally I would be asking you guys all sorts of Socratic questions, but I'm just going to lay things out here. So to convert a time domain to a frequency domain, we would use a Fourier transform, which is this incredible piece of mathematics. Fourier was this guy in the 1700s who came up with this brilliant idea of the Fourier transform, although it wasn't called that specifically until later, but it has applications in everything, in physics, mathematics, engineering, signal processing, everything from your cell phones to image processing and astronomy and all sorts of electronics. Everything has these creative and brilliant applications of Fourier transform. But in terms of EEG, all we're doing is separating out a signal through time into its component frequencies. So you can kind of think of it in musical terms as well. So when you hear a note, like let's say you hear a middle C pressed on a piano, you're not just hearing a pure sine wave at 262 hertz. That would be something more like a flute or something with a more pure, more true to form sine wave oscillation of pressure through the air. But when you hear a piano, there's more subdued and complex harmonics going on in the string of the instrument, and including the vibration of the soundboard and everything else. So every piano has a slightly different sound, and even the strings are designed to either enhance or minimize these sorts of harmonic oscillations. So the thing that makes a piano sound like a piano is actually a composition of multiple frequencies, even though the main frequency that you hear might just be that middle C note. And if you change instruments to a violin, that's going to have a whole different set of harmonics than a piano would have. And that would be different than any other instrument. So these harmonics are what create unique sounds of instruments. And if you isolate those harmonics with a Fourier transform by converting the sound in time into a separation of all its component frequencies, then you can actually see distinctly why one instrument sounds different than another. So that's kind of what we're doing here with EEG. We can break things down into their individual frequencies, and it just turns out that when you do that in the average person, you get this nice little layout of 5 different peaks of frequencies. So we've named the lowest band, we've named it delta, the next one theta, the next one alpha, the next one beta, to the highest frequencies that we get, we call them gamma frequencies. So for whatever reason, that's just the way the brain is constructed. And it has these bands of frequencies that tend to predominate. If we go through their unique sort of descriptive characteristics, delta frequency is about 1 to 4 hertz, and this can predominate in things like deep sleep or even dementia. The theta band is about 4 to 8 hertz, predominant in learning and exploration, like if you have a rat exploring a maze, for example, and REM sleep. The alpha band is about 8 to 12 hertz and predominates during difficult mental processing, during awareness, closing your eyes. Interestingly enough, although this doesn't occur in people who are congenitally blind, closing your eyes would be irrelevant, so they don't exhibit this phenomenon in general. The beta frequency band is 12 to 30 hertz, and this is more predominant during arousal and attention. And the gamma band can be 30 to 60 hertz, maybe up to 100 hertz. Some people say this is the sensation and perception rhythm. Some people have called it the binding rhythm. And it's all sort of research phase. And I used to think that it wasn't that meaningful or a bit of a stretch, but it's actually interesting because I saw this study in 2017 out of Germany that showed if you had reciprocal inhibition of 2 close proximity neuron tracks, it would naturally oscillate in these bursts of flow and rest. It would naturally oscillate at the gamma frequency range. And it's kind of complicated to explain. I'm not going to go into a huge amount of detail because you're not going to be tested on this. But if you're interested, you should look it up. The basic circuitry is just that you have 2 parallel pathways. And with reciprocal inhibition, it not only prevents crosstalk of the 2 parallel pathways. You can imagine if 2 neurons are running right next to each other, depolarizations in one could possibly trigger depolarizations, or at least easier depolarization of the other adjacent neuron. But with this sort of reciprocal inhibition, it prevents this crosstalk. And with this parallel processing, it turns out that it just naturally wants to oscillate in the gamma hertz frequency band. And so this sort of nice non-interference of simultaneously parallel flowing information through these pathways is a pretty cool example of how this could really be involved in a binding rhythm that enables us to make sense of multiple different pieces of continuously incoming information, congealing them into this one coherent consciousness of how the world is functioning. So, how do we make sense of all these cues, visual cues, sensory cues, auditory cues, positional cues, balance, everything? And how do we make sense of our interactions with the world and assemble it into one singular consciousness? It's a complicated question, or it's actually been called the hard question, so to speak. There's a lot more to this. So anyway, that's just an interesting side point if you're interested in pursuing more about that. So as far as clinical applications of EEG, we already saw examples involving epilepsy and brain pathologies. And it can also be used in a lot of research questions with things like visual evoked potentials or auditory evoked potentials. Some of my earliest research in undergrad was trying to sort out auditory neural networks and how the brain makes sense of sounds, how it's able to interpret things like harmony or dissonance using these auditory evoked potentials. But some other interesting applications include sleep studies, biofeedback, and neural prosthetics. So you can with neural prosthetics, you can put a sensor on the scalp, or sometimes they'll even put them deeper under the dura over the cortex. It's called electrocorticography. You can record these signals and try to develop algorithms to make sense of them. It requires a lot of training, obviously, on the part of the patient. But as an example, let's say someone has lost an arm and has a prosthetic limb. They can get an EEG sensor or an ECOG sensor, and over time train software and train their own brain to give certain signals that indicate desired motions. So if you have a thought of lifting up your arm in a bicep curl, for example, you could eventually program the software to trigger the robotics to move the arm in the same desired motion. So the signal would have to be both sensitive and specific enough to do the different specific desired movements, obviously. But it's a cool technology that's still being actively worked out. Okay, so also make sure you understand here how to manually transform a time domain to a frequency domain. Fortunately, you don't actually need to do this Fourier integral. It's pretty common sense if you just have simple waves to deal with. So, for example, just select the time interval that you're interested in. Let's pick a 1 second time frame. And if we have 3 equal oscillations in that time frame, then our predominant frequency is 3 hertz. So we can just chart in the Fourier graph in the frequency domain and make a peak at the 3 hertz value. So for our purposes, we can just keep the amplitude of that equal to the amplitude in the time domain. So it's all pretty straightforward. Okay, I should just point out another way to measure signals from the cortex is something similar called magnetic encephalography, or MEG, M-E-G. It's just a different way of sensing electromagnetic signals from the brain. But with this, typically action potentials cannot be picked up by MEG just because you have ion currents flowing in opposite directions across the membrane, sodium in, potassium out, and we don't have the temporal resolution to be able to capture the moving charges that are essentially canceling each other out. But we know from Maxwell's equations that whenever you have flow of current, you're going to have some amount of magnetic field, and typically you need on the order of tens of thousands of neurons to even make a detectable magnetic field. The main problem with MEG is both that we have lots of magnetic interference around us. So even though neurons are generating magnetic fields, there's so much interference from Earth's magnetic field and other signals that it makes it hard to filter. And there's also difficulty having a device that's sensitive enough to detect our own biomagnetism. So you can help that by using magnetically shielded rooms and improved detector technologies and things like that. MEG is able to detect intracellular currents at the dendrites of neurons. So typically, sensible signals are from neurons that are parallel to each other and reinforce each other's signals. So typically layers of pyramidal cells in the cortex, for example, are detectable with MEG. But getting into any deeper nuclei of the brain would be difficult. Although the signal is not going to be attenuated nearly as much by the bone and scalp. So in some ways, applications of MEG may actually be more interesting or more clinically relevant, but we're just not exactly sure how yet. So that's the basics of the lecture portion, and normally we would begin the experimental portion, like doing an EEG on yourselves and closing your eyes, having open eyes, doing mental arithmetic, doing hyperventilation, or holding your breath to see if any of that affected the EEG frequency bands. So typically what you would see in these Fourier transforms of your data is that during things like closing your eyes, you would see the peak of the alpha frequency band rising relative to the other amplitudes of frequency bands. So you would have to look at the Fourier transform and look at the significance of peak amplitudes compared to control groups of keeping your eyes open, for example. But like I said, due to this viral transmission going on, we won't be able to do that. But I hope this has been informative and at least helps you see some of the application and theory behind the application. So feel free to email me or comment if you have any questions, and I hope that helps. Thanks.